This video is on protein folding, and protein folding is a really important part of understanding biology because it really brings home the understanding of the way shape has such an influence on biological processes, and in particular complementary shapes and how they fit together. So folding of a protein is really about how the polypeptide chain, which you can think of as having a one-dimensional linear structure, folds into a three-dimensional structure. And we call the different ways in which it folds the primary, secondary, tertiary, and quaternary structure of that protein. But the 3D structure relates 100% back to the linear sequence of amino acids and how they interact with each other. So when we first make a protein, it's actually called a polypeptide. And that's about the peptide polymer that's made when you join amino acids together. So that happens during translation. You can see that page for that. But as it comes out of the ribosome, it starts to fold. And the reason is because the amino acid structure that we have here, each of those amino acids are chemical entities that have that amino end and the acid end that helps to form the polymer, but it has a side chain, and that might have different charges or different properties that mean it's going to be binding to or repelling other parts of the protein. And this is how folds happen. So the amino acid structure is called the primary sequence. Those amino acids will interact with each other at a local level to form some secondary structures. Key examples of that are here an alpha helix, but there's also one called a beta pleat, to form small local folds. The tertiary structure is then at the third level of how those folds and local structures form the three-dimensional shape. And then actually many proteins are made by putting together more than one polypeptide. It might be 4 copies of the same one that go together in a symmetry, or it might be different subunits of different peptides that go together to make a protein. And how they fit together is called the quaternary or the fourth level structure. So primary structure is the amino acid sequence, secondary structure, local features like an alpha helix, tertiary structure, the 3D structure of a single polypeptide, and quaternary is how they go together to make a protein. And this is 100% determined by those amino acid sequences and the environment that it's in. Because of course, the amino acid side chains are going to interact with each other, and you can see here 4 different ways that they can bond. So one is the very strong one shown here in yellow, is actually a covalent bond. So these sulfur can form a disulfide bridge where it's chemically connected to each other, and therefore this end of the peptide chain and this end won't come apart. But much more common is things like ionic bonds. So we have a positively charged and a negatively charged atom here, which are attracting each other. We have hydrophobic interactions. These two here in the grey don't want to be in the water, and so they will fold together to be with each other instead. And then here we have hydrogen bonds, just like the same hydrogen bonds that hold together the 2 DNA strands. Here we have a hydrogen bond holding together parts of this protein. And so all of these different interactions between amino acids will mean that depending on where they are and how they're located, that those parts of the polypeptide will fold together and form a structure. But all of that then depends on the solution that the protein is in and the temperature that it's at, because these charges will change depending on the pH, for example, and these hydrogen interactions too. So later on, when we look at factors affecting enzymes, I want you to link back and think about the protein folding, because this folding is what holds it together and keeps its shape that's so important for it to interact with other molecules, but the folding depends on the solution that it's into. That's covered in chapter 3. Protein folding and how it works is a major topic in biology to understand how we go from genes to functional proteins. Understanding a gene sequence only gives you some of the information. How that creates a protein and what that protein folds to and how it interacts with other molecules, that's really how you understand what the gene means. And this is covered in these videos here. Not really part of the course, but well worth watching if you're interested in protein folding.